cloud, and we don't have access to the cloud for now. So what we are going to do is very easy. Apparently, I could be doing it, but I can't be attending all the lectures. It's a bit hard. So you as individuals, if you're interested in a recording of a given lecture, I hope all of you are hearing me. You can hear me, right? So you as individuals, if you're interested in a given recording of a specific lecture, there are applications you download on your phone or the laptop that actually do a screen recording. I'm going to take you through one of them. Let me do my screen share and show you something. So you download them on your laptop, and once you download them, it doesn't matter whether you're the host or not. You can actually record what is happening on your screen. Are we together? So let me show you something in particular. If I can open it. Share. So there's an application called, I hope all of you can see that. You can see that? Can you see that? Are you seeing what I'm sharing?
Yes, you can see.
So there's an application called Format Factory. If you guys go, if you're using a phone, you can go in Play Store and look for Format Factory, and you look for that application. It can do a screen recording. So if you're interested in a specific lecture that is going on, you just, for example, this is the Format Factory. I've already opened it. So you literally come to someone has fallen. Are you okay? Need to maximize this. I hope all of you can see that. So for example, if there's a given lecture that is going on and you've opened your Format Factory, you just come and do a screen record. You see where my cursor is? You can see that. You can see that, right? Can you see that? I need someone to respond so that I'm not speaking alone. Okay.
Yes,
So for example, if a lecture has started, you just come and do screen record. And when you click on it, it's going to open and it will start recording your screen. You see that small thing? I don't know whether you're looking at what I'm having, but it will literally start recording your screen and you can go through it. Can you see what I'm having, or you can't see? Okay, so basically, let me just run back to that. I'm going to close this. Okay, so basically that's what I was telling you. You don't have to be the host to record your screen. You can download an app that does a screen recording. When you're doing a screen recording, it will take up all the PowerPoints that are going on. I can try and do for you a screen record for today, but I won't be able to stay up to the end of the lecture. But you have time now, you can do a screen record and get to see how it works for this specific lecture. A screen record will record the sound and everything that is happening on your screen. And if you are interested in watching that recording after the class, you can actually rewatch it. Otherwise, if I'm to do it for you, that means I have to be attending literally every lecture. And it is hard when someone is concentrating on the lecture and they're going to do a screen recording for you. That is going to be a bit hard. Yet it's a bit easier for you on the other side when you're attending the class and you just enable the screen recording as you're attending the class. I hope that is easy for us. Let's try it out so that you can these videos as fast as you can. We are sorry for not availing them. but we are having a challenge. So try and avail themselves. Okay, I'm giving the platform to the lady that is leading us today, Pauline Nalumaga. She's taking you through viral chemotherapy. I hope you enjoy the class.
Thank you very much, Barbara. Good afternoon, everyone. Am I audible enough?
You're very audible, Pauline.
Okay.
So I'm going to mute them for now so that they don't interfere with your class.
Okay. Thank you very much. Okay, good afternoon once again. My name is Nalumaga Pauline Petra, and I'm going to take you through viral chemotherapy. I'm going to take you through viral chemotherapy. So basically, in viral chemotherapy and in this lecture, we are going to try to understand the methods of controlling viral infections. Even right now, we know how the burden of this viral infection called coronavirus. So we are basically going to look around, we are going to try to understand the methods of controlling viral infections. And the common one is viral chemotherapy. Then we shall look at interferons and vaccinations. We are going to understand these more as we continue. So there is a viral chemotherapy challenge, and this is because viruses are obligate intracellular parasites. We know intracellular parasites means that they cannot replicate or express their genes without the help of a living cell. And this is the characteristic feature of viruses. They always multiply in living cells. So this makes formation of these drugs very difficult. And if you're to make an antiviral agent, it must be capable of selectively inhibiting viral functions without damaging the host. And also this development of drugs are very difficult because, you know, when a virus is going to replicate, it first integrates within the host cell. So you have to make a drug which specifically targets viral functions without interrupting the host the host cells. And another limitation during the manufacturing of antivirals is that many rounds of virus replication occur during the incubation period. And so the virus has spread before symptoms appear, making a drug relatively ineffective. Like in HIV infections, we realize that people start getting symptomatic when their CD4 cell count is really, really low. Then there is presence of several strains, including the current even coronavirus, where there are very many strains, and epitope variations. Epitope variations, it means this is a site on a virus on which a complementary antibody has to bind. So if you manufacture your drug, you're basically targeting a specific site. And when these sites change, leading to mutations, then the virus develops resistance. Some antiviral agents are active at a given pH and prevent infection within a given window. So if also the virus has a pH which is different from the antiviral drug that you have made, then the drug will be very ineffective. And we looked at it's very difficult to develop antivirals that can distinguish viral from host replicative processes, since most viruses integrate within the host cell. So before we even get to which stages have to be targeted, the stages of these antivirals, which stages of the viral replication they target, we have to first go back and understand the replication process of a virus. and I think we are now very well conversant with the replication process, where we shall have attachment, uncoating, early viral mRNA synthesis. Then we have early viral protein synthesis, viral genome replication, late viral mRNA synthesis, assembly, and release. That is a summary of the viral replication stage. Now, which stages are basically targeted by these antivirals? We look at attachment of virus to host cells, then the uncoating. These are the stages that aid in actually, these are stages that are targeted by the antivirus, the different stages that the various antivirals we have on market can target. We have the viral nucleic acid synthesis, translation of viral proteins, assembly and release of progenivirus particles. So this is a summary of the various antivirals we have. This image will make more sense after you have understood modes of action of the different classes of antivirals that we are going to look at in detail. Then after you've understood, you can just come back here and try to summarize the different processes here. So we have various drugs on market. These are some of the common drugs available in market. And this other column where we have the nucleoside analog, then a nucleoside analog is one of the classes, and not everything has to be a nucleoside analog. So we are going to look at the different classes. We are seeing like a cyclovir is a nucleoside analog, a metadinine is not. So we are going to understand more about this. This is also just a summary. Then the mechanism of action. Every class has a different mechanism of action, and the viral spectrum basically talks about which viruses, the drugs and the viruses that they target. So the first class, we have the nucleoside and nucleotide analogs. A nucleoside is, it consists of a nucleobase, which can be a purine or pyrimidine, a 5-carbon sugar. That is the nucleoside. While for a nucleotide, it still has a nucleobase, then a 5-carbon sugar, and it also has one or more phosphate groups. That is what differentiates a nucleoside from a nucleotide. Now, what do these analogs do? And they are the majority of available antiviral agents. So their mode of action is they inhibit nucleic acid replication by inhibiting polymerases, which are essential in nucleic acid replication. That is the mode of action. So these analogs inhibit cellular enzymes as well as virus-encoded enzymes. Now, that is a problem. So you have to get the analog which will specifically inhibit virus-encoded enzymes with minimal inhibition of the analogous host cell enzymes. Remember, we said one of the challenges is that antivirals cannot selectively act against virus-encoded enzymes. So virus variants resistant to the drug usually arise over time, and that is why we really have triple drug therapy. Basically, you have to use combinations of different antiviral drugs that can delay emergence of resistant strains. We've heard of the triple drug therapy that is used to treat HIV infections. So examples include, examples include of nucleosides. We have a cyclovir, lamivudine. I think we are more conversant with lamivudine, known as the 3TC. It's used zidovudine. And for nucleotides, remember we said for them, they have an attached phosphate group. And cidofovir is an example. Then the second class that we are going to look at is the reverse transcriptase inhibitors, where we have nevirapine, was the first member of this class. And the mode of action, it acts by binding directly to viral reverse transcriptase and disrupting the enzyme's catalytic site. We have to note the different modes of actions of these classes of drugs. And its problem is resistant mutants emerge rapidly. So the third class, we have the protease inhibitors, and saquinava was the first protease inhibitor, and it was approved in treatment of HIV infections. So these protease inhibitors, they are basically a peptidomimetic agent, and this means the peptidometric means a small protein like chain that is designed to mimic a peptide. So this agent fits into the active site of an HIV protease enzyme. And when it does that, it will inhibit viral protease, which is needed in the late stage of replicative cycle of the viral GAG and GAG polypeptide precursors to form mature virions, and activate the reverse transcriptase that is needed in the next round of infection. Therefore, all virus particles that are going to be produced are going to be non-infectious. And they include ritonavir, indinavir. Then we have fusion. The fusion is a large peptide that blocks the virus and cellular membrane fusion step involved in entry of HIV-1 cells. Then we have synthetic amines like amantadine and rimantadine. These ones inhibit influenza A viruses by blocking viral uncoating. Then we have foscarnet, which is an organic analog of inorganic pyrophosphate, and it selectively inhibits viral DNA polymerases and reverse transcriptases at the pyrophosphate binding site. Then we also have methicillin. This one was mainly used in poxviruses, and it also contributed to the eradication of smallpox. And its mode of action, it blocked a late stage in viral replication. Therefore, non-infectious virus particles were formed. The problem here, viruses, especially RNA viruses, mutate at a high frequency. At least that one, by now we know that RNAs mutate more than the DNA viruses. So this means that genotypes encoding drug resistance always arise rapidly. You know, these RNAs, viruses, their polymerases make more mistakes because they lack the exonuclease proofreading activity. So these drug-resistant genotypes may be at an advantage in hosts where the drug is present and may become the dominant genotype in these hosts. Now, there are various strains that have been linked to resistance, and these are the genes that have mutated against the various antivirals on market. So we have interferons. We have interferons. Interferons is another method of controlling viral infections. So interferons are host-coded proteins that are members of the large cytokine family and inhibit viral replication. We are going to understand more about interferons when we start with immunology, we shall really dig deep in the functions of interferons. So they are produced very quickly within hours in response to viral infections or other inducers, and are one of the body's first responders in defense against viral infections. So they are central to the innate antiviral immune system. We shall appreciate more the innate immune system when we go to immunology. And they modulate the humoral and cellular immunity and have broad cell growth regulatory activities. So how do they do their antiviral activity? They have multiple species of interferons, and they fall under 3 groups, which is the alpha, beta, and gamma interferons. And interferon alpha and beta are considered type 1, or they are also known as the viral interferons, while the gamma is known as the immune interferons. And they are detected after the viral infection in intact animals. So what interferons do, they will not protect the virus-infected cell that will produce it, because it's not itself an antiviral agent. But then when it is produced by this virus-infected cell, the interferon will move to other cells and induce an antiviral state by prompting the synthesis of various proteins that will actually inhibit viral replication. And these interferon molecules bind to specific cell surface receptors on target cells of these viruses. So receptor binding will trigger triosine phosphorylation and activation of transcription factors in the cytoplasm, which will then translocate into the nucleus and mediate transcription of interferon-inducible genes. Please, in case you do not understand anything, feel free to ask questions. And we also have to read more and understand this, and it will even make more sense when we get in immunology and get to study interferons more. Then there, when you relate its function, you will understand its antiviral activity more. So the antiviral state acts selectively against viral mRNAs and not cellular mRNAs. There are also various steps in viral replication which will be inhibited by interferons. So these interferons are not always host species specific in function, but they are almost always host species specific in function, but they are not specific for a given virus. So when interference is added to cells before infection, there is marked inhibition of viral replication, but nearly normal cell function. Because they are specific, they will only target viral mRNAs, but not cellular mRNAs. So they will not interfere with the normal cell functions, unlike various antivirals that we have on market that interfere with the normal cell function. And they're extremely potent. So we have looked at interferons, and they have a very interesting way of trying to fight these viral infections. However, every microorganism always comes up with mechanisms on how to counteract the immune system or to counteract any method that is being used to suppress it. So we are going to look at the virus mechanisms that are used to counteract the effect of interferons. So we have specific viral proteins may block induction of expression of interferons. Then they may block the activation of the key PKR protein kinase. This protein kinase R plays a very major role in centrocellular processes like the mRNA translation. So it is very important even in the manufacturing of the production of interferons. Then we have these viruses may activate a cellular inhibitor of PKR, this protein kinase R protein. So they may also block interferon-induced signal transductions. And remember, if interferons are not produced, therefore, these cells will not have, the cells of a host will not have antiviral properties. So that means viruses can luxuriously multiply in the host. Or they may neutralize interferon gamma by acting as a soluble interferon receptor. So we have looked at the viral chemotherapy where we looked at different drugs, different classes of drugs. Then we have looked at interferons, and we have seen interferons they are produced within our bodies to try to fight infection, and they are in the innate immune system, which we shall appreciate more when we start with immunology and we look at the innate immune system. Now, the last method of controlling viral infections are viral vaccines. So what are the general properties of viral or the general principles of viral vaccines? The purpose of viral vaccines is to use the immune response of the host to prevent viral disease. Several vaccines have proved to be remarkably effective at reducing the annual incidence of viral disease, and vaccination is the most cost-effective method of prevention. Remember, we know prevention is better than cure. So it's better to prevent before you start investing a lot in these antivirals. And immunity to a viral infection is based on the development of an immune response to specific antigens located on the surface of virus particles of virus-infected cells. So in this picture, we are looking at the examples of approved virus vaccines by 2011. So we shall have the common vaccines. I think we know of hepatitis A. So these are the vaccines. We have vaccines for hepatitis A, B, influenza A and B. We have measles, mumps, papilloma, poliovirus, rabies. We have the rotavirus. We have rubella, varicella, zoster, and of course now we shall add on corona. This was for 2011. And we have different types. The types that we have are the killed vaccines that we are going to look at, and the subunit. Subunit is basically also like a killed vaccine, the killed type. We shall also look at it in detail. And then we have the live vaccine. Then the cell substrate means where these vaccines were manufactured from. In the human diploid fibroblasts, we have the yeast embryonated chicken eggs, monkey kidney cells, and human diploid fibroblasts. So the killed virus, that's the inactivated virus. Sorry, vaccine. These inactivated vaccines are made by purifying viral preparations to a certain extent and then inactivating the viral infectivity in a way that does minimal damage to the viral structural proteins. And mild formalin treatment is often used. So basically here, it's a virus that after purifying and you get the virus itself, then you try to inactivate it, you try to kill it, and we use mild formalin. So this vaccine will have the intended virus but it will be killed, and it will be enough to elicit an immune response. So these vaccines are prepared from whole virions and stimulate the development of circulating antibodies against the coat proteins of a virus, conferring some degree of resistance. And they have different advantages. The first advantage is, you know, that this virion, the virus, has been killed. So you know that you will have no reversion to virulence by the vaccine virus because it is dead. So you know that it has died, therefore there is no development of disease. And vaccines can be made when no acceptable attenuated virus is available, but they also have disadvantages. So the first disadvantage is you have to be extremely careful when manufacturing them to make sure that there is no residual live virulent virus, because if there is any live strain, then that means you're just injecting viruses in people, and instead of saving lives, you're going to be destroying lives. And the immunity conferred is brief and has to be boosted, which not only involves logistic problem of repeatedly reaching the persons in need of immunization, but also has caused concern about possible effects. You know, when you get a vaccine, there are various effects. Now imagine every booster dose you have to get these hypersensitivity reactions. And parenteral administration of killed virus, even when it stimulates, it does stimulate circulating antibodies to satisfactory levels, but also it gives limited protection because the local resistance IgA is not induced. So that is about the killed virus vaccines, and the cell-mediated response to inactivated vaccines is generally poor. Now, before we look at the attenuated live vaccines, remember in our picture, the other side, we saw subunit vaccines. So subunit vaccines, by the way, it's just a class of inactivated whole cell vaccines, but for them, they don't contain the entire virion. They just contain part of a virion that is necessary in eliciting an immune response. So a subunit is just like a killed vaccine. A subunit vaccine is just like a killed vaccine, but for it, they don't use a whole virion. They only use specific parts of a virion. Then we have attenuated live virus vaccines. So in these live virus vaccines, they use virus mutants that antigenically overlap with a wild type virus, but are restricted in some step in the pathogenesis of a disease. Advantages of live vaccines, live virus vaccines. They have an advantage of acting like a natural infection since it's just a mutant of an actual pathogen. So that means if one is injected this vaccine, you're going to have the same immunity since it's going to act like the actual infection that you would have had. And they multiply in the host and tend to stimulate longer lasting antibody production. They induce a good cell-mediated response, and this is because they mimic the natural infection, and they're even able to multiply in you. Therefore, they will induce antibody production and resistance at the portal of entry. But what are the disadvantages? Remember, they just mimic, this is a mutant of the actual infection. So there is a very greater chance of this mutant to actually go back into the other strain that we are trying to avoid. then it will cause an infection. So this is what the first point says. The risk of reversion to greater virulence during multiplication within the person who has been vaccinated. Then these mutants are basically the cell substrates used in manufacturing of these vaccines are mainly eggs, primary cell cultures. So these agents in the culture substrate may enter the vaccine stocks. And the problem of these contaminants, they may be circumvented through use of normal cells serially propagated in cultures. The storage and limited shelf life of attenuated vaccine presents problems. So that is why various vaccines, we get them viral stabilizers to try to keep them, for them to be effective. Then they require cold chain storage, and there is interference of co-infection, hold on. Interference by co-infection with a naturally occurring wild-type virus may inhibit replication of the vaccine virus and decrease its effectiveness. So you have to be very careful in a very sterile environment, because if there is any virus that is going to come, there will just be competition of substrates, and what therefore it shall not replicate at the level that we want it to replicate, so that when it's injected in you, it can actually be of importance. It can elicit that immune response that we want. So these are comparison between the killed and live virus vaccines. We've looked at the killed vaccine, you need multiple doses, while for the live, you just need a single dose. And for the killed, you need an adjuvant. And duration of immunity, with the killed, we looked at it needs booster doses, therefore the duration of immunity is shorter. Effectiveness of protection, we looked at the live vaccine offers more protection than the killed vaccine. Immunoglobulins produced for killed, it only produces IgG, but for live, it produces the IgA and IgG. These comparisons are, this slide is very important, because it is very important for us to know and appreciate the difference in the killed vaccine and live vaccine. You have to know the number of doses. It will just make you appreciate vaccination more. Then we have the cell-mediated immunity produced. It is poor in the killed, but great in the live vaccine. Residual virulent virus in vaccine. It is possible, if the manufacturer is not careful, these residual virulent virus strains can actually be transported or can be passed on in the vaccine. Reversion to virulence, it is not that can revert to go back to what we are trying to avoid. Excretion of vaccine virus, it is possible in the live, because this thing keeps on multiplying even within the host. Interference by other virus in the host, it is possible in the live vaccine, but not in the killed vaccine. And we know live vaccines are not stable at room temperature. They need a specific temperature for them to be kept moving. And that is the end of today's class. All questions are welcome.
Hello.
Yes. Hello.
About this, the attenuated. So does it mean that they also multiply within our bodies in order to evoke the response of the body? Or they, as in the culture, they first multiply in the culture, then as in the body, their number is constant, or they also duplicate in our body?
They can, They can, but no, it's possible. It is just possible, but it's possible that they can multiply in our bodies. And that is why you just need one dose for them.
Hello?
Hello, can you hear me?
Yes, we can hear you.
Yes.
I want some explanation on adjuvant.
Adjuvants are, these are, what can I say? Like, they are used to make the vaccine produce a bigger response, because you have seen in the killed vaccines, they might not elicit a proper immune system that we want. So these adjuvants are added, like some aluminium salts. They are added in these vaccines. Since also now we have even seen the subunits, you're just using a very small portion of the germ of the virus. So it needs a vehicle. Actually, an adjuvant is like a vehicle. It's like a vehicle that is going to enable this vaccine work more to elicit a better and bigger immune response. Has it made sense?
Yeah, excuse me, madam.
Yes.
Thank you for the presentation.
You're welcome.
Mine is a smaller addition on
Yes.
amantadine, much as it is an antiviral drug. Can you hear me?
Yes, I can hear you.
Yes, much as it is an antiviral drug,
Yes.
it has been used in the management of Parkinson's disease. Because
oh, yes, it has.
Because when it crosses the blood-brain barrier, it acts as a dopamine agonist. I submit.
Thank you very much. Thank you very much. Any other additions or questions?
Excuse me, madam.
Yes.
I hope you can hear me.
Yes, I can.
Mine is it's also a supplement on the live attenuated viruses, and maybe it can help us to understand something better when we put it in perspective. When we look at the oral polio vaccine, it's a live attenuated vaccine, and previously it used to be containing all the strains of poliovirus 1, 2, and 3. But due to what you've just told us, something called reversion to virulence, it was observed that strain 2, the poliovirus 2, was having a great tendency of reversion to virulence. And in the recent vaccines, it has been removed. And now OPV, the oral polio vaccine, contains only strains 1 and 3 for that reason.
Yes.
Yeah, so I was just trying to put it in perspective.
Thank you very much for making it more practical.
Thank you too.
Thank you very much. Any more additions or questions?
Excuse me, madam.
Yes, madam. Go ahead. I beg your pardon.
Thank you so much for the presentation.
You're welcome.
Yeah, I would like to inquire what kind of vaccine is the current COVID vaccine they are using? Where does it fall?
Now, for the current COVID-19 vaccines, it is a bit tricky because there are various vaccines, right? There are various vaccines. So we have the mRNA, some of them are mRNA, and I have not yet really got enough knowledge about where these vaccines, by the way, fall. That's the truth. Because when you try to look more about it, you really don't get enough information about it. And I'm trying to get information from the World Health Organization and CDC, but I have not yet got enough knowledge about them.
Okay, then the other question is like, how long does it take for someone's immune response to be elicited? Because people are receiving, for example, the COVID vaccine, and they are told they still have to wear their mask, and they give an excuse that it is because the immune response is not yet activated, or it is not yet elicited. So I would like know how long does it take after someone has received the vaccine?
Actually, for this COVID-19, we are basing on what they keep on telling us. So I think that was last week or the other week. Anyway, the last time that I was reading about them, I've told you where I get information from. They say that it takes 2 weeks for you to get this, to build up your immunity. However, COVID-19, it's very tricky to make its vaccine. Remember, it's an RNA virus, and we have looked at them mutating rapidly. And then there are so many strains that are coming on. There are so many strains of COVID-19. When they just developed the one for the alpha strain, we got to see that the beta one came up. There is the gamma. The lambda is almost being, the lambda variant is almost being declared. There are so many variants, and that is making it hard for them to make a vaccine. That is why the vaccines that have been made for COVID do not protect you 100%. So even when you get this vaccine, you still have to wear your mask, because you can get another type which will not be stopped by the vaccine that you have got. That is the problem. So we need to read, we as future virologists, we have to look at a way of how we shall incorporate all these mechanisms viruses use to dodge methods of controlling them into winning them. So that is the challenge. Otherwise, when you get vaccinated, your body has to produce antibodies, because it has entered it. So when it recognizes it, it still has to produce antibodies to protect you as soon as possible.
Okay, thank you so much, madam.
You're welcome. So any more additions? Any more additions? I have unmuted. I think you can now ask the questions. I have tried to.
Excuse me.
Yes.
Mine is a question about interferons. You said they when they are produced, they activate the cells so that they get ready maybe for incoming virus. So can we use that mechanism to prepare all our body cells for any form of incoming virus, either they comfort us protection against them? And if so, can that be considered a vaccine?
Oh, by the way, interferons are within us. They are within us. It's
But
like how you. Yes.
Can they be manufactured maybe from a pharmaceutical factory then that injected into our bodies?
As a future virologist, you will join that team that is trying to do that because they are being studied.
But I think they are available. We have.
Yes, they are, in that they are trying to even see if they can be necessary in treatment of COVID-19 vaccine. COVID-19 patients. So, yes.
Would that be considered a vaccine also?
It won't be considered a vaccine, but it is a therapy.
Okay, thank you.
You're welcome.
Excuse me, doctor.
Yes.
My question is, why is it that in the live attenuated we have IgA while in the kill don't have it?
So it's just because it does not elicit it. It does not. It's immune system. The killed does not produce. That's why it needs to be boosted. Like that is one of the disadvantages. It's just one of the disadvantages. It just can't produce it.
Excuse me, madam.
Yeah, I'm sorry to interrupt. So I'm sorry my environment doesn't allow me to, so I'll be a bit quick. She's really, really clear on that. Like we're going to have the immunology lectures. You're going to meet IgA. It falls a bit under innate immunity. And something interesting about innate immunity, this body recognizes the virus when it is coming in. It is the first line of defense. That is where IgA falls. It's the first line of defense. Yet when it comes to adaptive immunity, it a little bit dwells on memory. So the fact that we have IgA, when we cover these antibodies in immunology, they're going to cover whatever questions you're having now. So we have IgA falling into innate immunity as a first line of defense. Now, a live virus, normally it behaves almost like when the virus enters the body. So the body recognizes this as foreign. While for the killed virus, there are things it doesn't express like the live virus expresses. That is why we have to boost it with time, because it doesn't behave like the live virus. These things will make more sense when we reach immunology, like in the previous lectures when I was telling you, let's stay in virology a bit, because immunology is going to be more fun for the questions you're going to be having. I really can't stay unmuted for long. My environment won't allow me, but let's not cut immunology. I'm going to do these classes when we reach the immunology lecture.
All right, thank you.
You're welcome. So, any more questions?
I think that, okay, Pauline. Madam Pauline, thank you so much for the lecture. So someone is asking, why didn't immunology come first? Sorry about that, no, miss. We normally start with virology. Immunology will make more sense the first that benefit. You guys, why do you use names that don't belong to you? You guys want to unmute? Let's try. We normally mute you guys because you misbehave when we unmute you. So that is why we mute you. But you can unmute to ask your question. So I'm going to answer someone. Was it your miss? We normally start with virology for some of these things to make sense. But now immunology will come in so that you get to know, to connect the things. These things, they run hand in hand. And it is one specific semester. It's not like we split them in different semesters. That is why in the previous semester, you have virology and immunology. They run hand in hand. They will make more sense. The good thing is you're starting immunology next week. That takes me to the point. If you look in that timetable, You're going to notice that next week on Friday, you're supposed to be having a practical, which we won't be able to have because of the environment we are in. So what we are going to do, there's a class we're supposed to have with me, but I'm going to be taking you through innate immunity. I'm literally, someone is going to give you an introduction on Wednesday, and I'll take you through innate immunity. But you're going to appreciate and can't hold your thing. You're going to love it. So I'm kindly requesting you, Thursday, the fact that we have Friday, which is scheduled for the practical, and we're not going to have the practical. So the fact that on Friday, we are supposed to have the practical, and it is not going to be there. I'm kind of requesting that the class of Thursday, we schedule it to Friday, since the Thursday time is a bit little. It's really, really little, and that innate immunity covers a lot. So I'm sorry I took you in a bit earlier, but I'll keep reminding you even in the weeks to come. In the week to come, that the Thursday lecture will be on Friday, since Friday has more time than Thursday. But the Wednesday lecture will be in the right time, but the Thursday lecture will be on Friday. Okay, I've really interrupted Pauline's session. So thank you so much, Pauline, for the lecture. People, make sure you register, and let's meet again next week on Wednesday. Thank you so much.
Thank you so much. Have a good evening.
Thank you very much, Madam Pauline.